Perfect. I think we are live and we are ready to go. So, hello everyone. I'm Amrita Jain. I'm Investment Director at DeepRight, and I'm really excited to welcome you all in today's webinar. We are going to dive into something really important for all of us, and that is longevity and healthy aging, and how science and biology and technology can change the way we think about it. So this today's session is hosted by Hello Tomorrow and L'Oreal. Just a quick word about Hello Tomorrow. It's a global group that support deep tech startups solving the real big world problems from health to climate to energy and more. And at Deep Right, we invest in early stage companies. We are powered by Hello Tomorrow, and we like to invest into deep tech companies who is using deep science and engineering to solve global challenges in health, sustainability, and beyond. And today, we will explore the science behind healthy aging, and in particular, how L'Oreal is thinking about it with their new innovation called Cell BioPrint. So before we could start, there are some housekeeping informations. So this webinar will last for 1 hour, 45 minutes for discussion, and the last 15 minutes for question and answer. So please do send your questions in our Q&A box. You can find this in the right bottom. This webinar would also be recorded, so it will be available afterwards. So now let's start. So I'm today joined by a fantastic panel of experts from L'Oreal, and I will let them introduce themselves. Maybe we could start with Carine.
Hello, everybody. Nice to be with you today. So I'm Carine Ballihaut, working for 21 years at L'Oreal. I'm a scientific transformation director within the scientific direction at L'Oreal in advanced research, and especially working on the ingredients and on the territory of longevity. Nice to be with you today.
Oh, thanks. Maybe Nukhet?
Yeah, hi everyone. My name is Nukhet Cavusoglu. I've been working at L'Oreal for 20 years, almost 20 years, in advanced research as well as Carine. And I'm the head of a platform which is named High-Throughput Biology and Diagnostic that deals with, I would say, omics technology to decipher biological mechanisms in our favorite tissue, which is the skin. And the topic of today, for sure.
Thanks, Nukhet. Fred?
Hi, I'm Fred. I've been at L'Oreal 12 years, so I'm just a baby at L'Oreal, as you can see, compared to everyone else. I am working in our beauty tech team, leading tech innovation out of the US. So normally based in New York. I'm in Paris at the moment, but normally based in New York. And what we do is we look for the intersection of beauty and tech, whether it's devices, tools, skin tools, hair tools, diagnostics. So it's the topic of today. We also do algorithms, digital, so anything beauty plus tech. And my team works with Carine and Nukhet and all the scientists at L'Oreal to bring kind of the most cutting edge science to the consumer through tech, where we have the opportunity, which we did for this project.
Perfect. Thank you so much for your kind introduction. So now let's get started. My first question could be Carine and Fred. So very big question, what is longevity? It could mean different to different people, but what does it mean in this context and to L'Oreal?
Okay, so maybe let's start by understanding the roots of our discussion, because longevity is a fascinating concept that has inspired frescoes and novels for centuries, from mythology to science fiction. But it was only in the 90s that it really emerged from a scientific standpoint, with the discovery of genes that have an impact on aging. Then it has been clearly crystallized and defined in the literature by Giovanna De Benedictis and Claudio Franceschi in 2006 as being the average lifespan under ideal conditions, with the ability to live a long and healthy life. This concept can, of course, be applied to how all our tissues including skin and scalp, the subject for L'Oréal. And when we consider longevity from a dermatological and medical perspective, it involves enhancing novel strategies to counteract intrinsic aging mechanisms, prioritizing health and promoting resilient skin. As we fast forward to the present, the topic of longevity has seen an exponential rise in interest. Between 2000 and 2023, publications increased more than 12 times, with nearly 100,000 publications dedicated to longevity. Notably, 10% of these publications explored the intersection between longevity and skin, with nearly 10,000 publications. This rise indicates a growing awareness of the importance of understanding and addressing aging mechanisms. Those numbers not only signify a substantial increase of interest, but also a vast landscape of scientific opportunities and possibilities for us. Now, let's delve into an important point of our discussion: the wheel of longevity. Maybe you have heard of it. Initially presented by Carlos and Houtin and Hall in 2013, and later refined in 2023, this conceptual framework outlines 12 hallmarks of aging. Around the wheel of longevity, each hallmark provides invaluable insights into the aging process, presenting us with potential targets of intervention. In essence, these 12 hallmarks provide a roadmap to understand aging at a cellular level. They guide us in comprehending the interconnection mechanisms of aging. Researchers hope that by understanding these factors, intervention can be developed to slow or even in the future reverse the aging process, improving health span. This framework of 12 hallmarks does help us direct the translation of longevity to skin through invaluable biological insights into the aging process at the cellular level. It's important to understand that these hallmarks in reality are interconnected, and we have to understand the different hallmarks and their connection to really understand aging at a cellular level. It is very complex. The breadth of hallmarks also presents us new potential, new targets, new approaches for intervention that can slow the aging process and extend the health span of our skin in a connected way that delivers the best care for patients concerned.
Cool. Thank you so much. I mean, it's impressive. 10,000 publications so far. Great. Nukhet, we have seen a lot of buzz around longevity as well, from supplements to wearable to cell therapy. Can you give us a quick overview of main approaches out there? What's the bigger picture look like?
Yes, of course. I want to say to begin that the establishment of this longevity wheel was founding for every scientist to align on those hallmarks and find new intervention, therefore, to address the root causes of hallmarks. So the root causes of aging. So this is the first approach that deals with intervention. So here are highlighted a few of them. And largely inspired from the medical field, actually. So you have metformin, for instance, rapamycin, molecules very well known in promoting the healthy lifespan. And there are many other, and I can cite, for instance, the targeting of senescent cell, which is a hallmark of aging, the targeting of senescent cell with what we call senolytics that will kill those senescent cells, also known as zombie cells, or even developing xenomorphics that doesn't kill the senescent cell but target the harmful factors secreted by the senescent cell. So there are many examples that I won't cover here, but just let you know that there's more than 100 clinical trials ongoing on those interventions. And what this shows is the fact that there's need of evidence as well in this emerging field of longevity. So this was for the first approach. The second approach, which is a general one as well, is the development of omics technologies to explore the field of longevity. So this is mainly due to I would say, the big advances in the last decade on specific technologies such as the next generation sequencing and the mass spectrometry, the high resolution mass spectrometry technologies that both reach an unprecedented level of high throughput as well. So to generate those massive data, it was very important to generate those massive data, I would say. So here, the main omics with, as an example, so genomics. Genomics is, in the context of longevity, what we look is in the genomics, the gene variations, the variants that are in the genes that are linked to premature aging, for instance, or to your susceptibility to an age-related disease. There is the well-known epigenomics that you all heard about, is sequencing, is dealing with the measurement of DNA methylation. And those DNA methylation, we are known that they are sensitive to our exposure and our lifestyle. So following those markers through epigenomics is of interest for sure. There is transcriptomics as well that measure the level of the gene expression. And this is a very important thing to do when you see the up and down regulation of the RNA. Then you can have information on the pathways that are activated, activated during aging, for instance. And then there are two other omics that are more, I would say, downstream omics, proteomics and metabolomics. So proteomics is really interesting because it's my favorite one. It deals with the characterization of all the proteins within a system. And by doing proteomics, you can get the identification of biomarkers of interest, of targets that can be actionable through with drugs, for instance. So proteins very important, and moreover, it can give you, well, interesting biomarkers in the context of interest for us, in the context of aging. The same for metabolomics. It deals with addressing small molecules, metabolites, that are involved in all the metabolic process within your cell, so very important as well. And you can follow the metabolites evolution within the system and get information on where you are in terms of your age trajectory, or monitor sometimes the effectiveness of interventions. So overall, to conclude on this approach, omics give access really to at a molecular level to the mechanism that drive your aging process. So what is important here to note is that it can be totally individual. It is your aging, your molecules that gives you your biological status, a very global one, which makes you unique, and ultimately could monitor your personal aging trajectory, I would say. So this is it for the two approaches.
Perfect. Thank you so much, Nukhet. My next question could be to Carine. I mean, you have been seeing this field evolve over years. So what do we understand about how skin ages on a cellular level?
Yeah, for sure. So at L'Oréal, longevity is a research focus for our experts for more than 20 years, in fact, across different and connected hallmarks. I didn't cite each of them, but you have a microbiome, you have epigenomics, you have mitochondrial dysfunction, and so on and so on. But these different hallmarks, and I insist, they are different but connected. And as leaders in skin biology, alongside the complexity of longevity, we believed it's more important than ever to be grounded in science and take an evidence-based approach to this topic. And therefore, we did many publications on the different hallmarks, individually ones, in fact, to be recognized by our peers. For example, we have many papers on photoprotections, 10 papers, for example. Not 10 papers, more than that. We have many papers on the microbiome, on the photoprotection, some of them on senescence, and we have a lot of manuscripts written in 2024. We have we wrote, in fact, on this field of longevity, globally speaking. It's very important for us. And as an example, I can take the impact of UVA damages, because these rays affect many of the hallmarks in the wheel of longevity, and we are working on it since many years. So in the wheel of the longevity, first of all, it can affect genomic instability, mitochondrial dysfunction, inflammation, senescence, proteostasis, and we have been searching the effect of the sun on the skin since 1938. That is to say, near 100 years ago.
Cool.
Maybe I can talk also about the ingredients we think about when we speak about longevity, because we are focused on the cellular aspects of aging and health, and we use many technologies to target cellular mechanisms alongside the wheel of longevity. But it's very difficult to translate them into clinical benefits. We see some familiar ingredients here. For instance, niacinamide, retinol, vitamin C, or resveratrol. But we also know that prevention needs to be a cornerstone of any longevity intervention. And sunscreen and environmental protection are well studied. Holistic solutions for patients, not only to prevent skin cancer, but also to prevent and to support skin's longevity mechanisms and protect against premature aging of the skin. I've got, for example, also an history, in fact, of the sun protection. And you can see it begins at the beginning of the 20th century. We have known that many years that UVA protection is essential to protect genes and overall cellular health, regardless of the time of year or climate. More than 30 years ago, at L'Oréal, we introduced Mexoryl SX, a major innovation in UV filter technology, providing enhanced UVA photoprotection. We have continued to advance our research on photobiology, focused recently on the long UVA, in fact, the most insidious rays. The newest filter, Mexoryl 400, isn't yet sold in the US today, but it's a good tool to illustrate how broad spectrum protection can help preserve longevity. Another tool in the prevention is the interception of reactive oxygen species before the damaged DNA and other core mechanisms of cellular function. We know that modern external aggressors, such as pollution particles, for example, and ozone, can be activated by solar radiation. They are photosensitive, in fact. And they can create this oxidative damage. A lot of our research on vitamin C and resveratrol, for example, work along the spectrum of longevity from this perspective, both within the skin and on its surface, to help preserve cellular integrity. I think the two are both interesting examples to see how we can promote longevity for skin health and for the organism.
Perfect. Cool. Thanks, Carine. Maybe Nukhet, I mean, you have been working on identifying specific biomarkers in the skin. Can you share a bit about what you're looking for and how and what they can tell us about skin aging and skin health?
Sure. So first of all, I would like to show you, before entering in the detail of our biomarkers, just to talk about this concept which came with longevity, the clocks, the biological, the aging clocks that are really more and more talked today and worked today. So remember, our identifying the right biomarkers is essential for sure, and having omics to characterize those biomarkers is essential as well. So this is why there are many aging clocks that were used from multi-omics dataset. As you can show here on the right panel, there are many studies published on different aging clocks obtained from different omics and biological metrics. And the most famous biological clock actually is, the epigenetic clock from Horvath, which was the first one actually more than 10 years ago that was established based on the measurement of your epigenome. And so these epigenetic clocks are very interesting and useful to predict a mortality risk, for instance. But after that, more and more, there have been more organ-related aging clocks that were generated. As an example, the brain was used, the omics from brain was used to predict the cognitive decline, for instance, or the heart used for predicting coronary heart disease, etc., etc. So the skin is the forgotten one here and is the missing one. So this is why we built, I would say, our own clock, aging clock. So to come back to the notion of aging clock, there is a scheme here very easy to understand on the left panel, where you see on the x-axis the chronological age and y-axis the predicted biological age with your omics, with the omics metrics, whatever the omics you use. So it can place you on this curve. If you're an average person, you'll be your chronological age, which is the age on your ID card, would be equal to your predicted biological age. If you're above this curve, then you are an accelerated fast ager, I would say. And if you're below this curve, in the cloud below, you are a slow ager. And this is defined with some biomarkers of interest. So this is the approach we used to work on our biomarkers. And here is an example of our last study that delve into those specific biomarkers that were identified on skin. So it's a proteomic work. You have, because we have more 20 years of knowledge on proteomics applied to skin. So in the study, we studied 351 women aged from 18 to 75 years old, and we assessed 21 clinical signs by expert assessment, and we did a lot of statistic in this study to clusterize those clinical signs, trying to understand their distribution among ages and among this population. And we measured for each of these women 5 proteins. So those proteins were shortlisted from our previous knowledge on skin proteomics, and we used those 5 proteins to predict their, I would say, their trajectory in getting some clinical signs that was unique to them. So we have shown that many correlation, in other words, between the level of those proteins and their clinical signs, and being capable of doing that to predict what clinical sign they are prone to develop in the coming years. And in addition to that, we have built an algorithm which works with the level of those proteins that can predict either an age acceleration or deceleration for this person. And what we have shown here is that 3 out of these 5 proteins are predictive and are the most predictive ones, actually, for age acceleration or deceleration. So to make a long story short, all this knowledge and expertise has been translated into our innovation directly for our consumer on point of sale, and this is the Cell BioPrint.
So, thank you. I mean, the goal is definitely to stay below the curve and
Exactly.
and age later and better. Fred, all the research that Nukhet just described, how does it lead to Cell BioPrint? I mean, what exactly is a Cell BioPrint and what does it do?
Definitely. So I think 2 things first, kind of to introduce that. One, with beauty tech, we're always looking for ways to bring what our scientists and our biologists and what our experts know about skin and what they do in the lab to the consumer. So you've just heard about all of these biomarkers, which we know are indicative of different skin signs and the trajectory of aging, and we use in product development. So, but we've never been able to bring them to the consumer before. So the techniques that the team has used are very much lab techniques that use take time and use multiple steps to process these readings. At the same time, in the space of longevity, I would say diagnostic tools become even more important. So when we think of skin diagnosis, the most basic thing is, what is my skin type? And maybe maybe like intuitively or maybe an expert told you. But then it gets even more detailed of, what degree of wrinkles do I have? Are they getting worse? And these are things we can measure optically, visually, and use even AI algorithms to measure. But when it comes to longevity, if you do longevity interventions correctly, you won't see anything. So like if we can treat skin aging from a longevity perspective, you won't see the aging. But how do I know if that's working? How do I know if the intervention I've done is working? And what intervention do I need to know? So these types of biological driven diagnosis are, we think, incredibly important for longevity. And when we get these new longevity treatments, it's going to be super important. And bringing this research to the consumer is a huge goal for us. So that's where L'Oreal Cell BioPrint comes in. What we did here is we were able to take a technique that uses microfluidic lab on a chip technology with a device that can be installed at different points of sale. So at department stores, at boutiques, at medi-spas, and at dermatologist offices. So it can be right there at the place where the consumer is already coming. And with a super simple skin sample, we can tell you the levels of the actual biomarkers, the actual proteins that Nukhet was talking about. So all you do is, this is the device that you see here. You take a piece of tape, pretty much a tape strip, a D-squam, and you stick it to your cheek, and you stick it until you have an entire tape strip full of pretty much dead skin cells. And these dead skin cells have in them the proteins. So I put that sample in a little, we call it a protein extraction device, but it's a little plastic cartridge with a buffer solution, similar to when you do kind of any kind of biological test, a buffer solution that breaks apart the cells, leaving behind a fluid filled with the proteins. And it just takes one drop of this on our chip. And what this lab on a chip cartridge does is it pulls the proteins through, and these channels are so small, the cartridge itself is etched with the same tech used to etch, like low tech, but to etch computer chips. And it pulls them through in such a thin layer that as they pass over, the proteins bind with antibodies, and the antibodies bind with fluorescent beads. And now you have this solution of fluorescent paired proteins that can be read by the machine. So it's a complex biological, but pretty simple to explain technique that lets us read right there in 5 minutes how many proteins you have. And what we're doing with that is right now 3 things. So this is how you would see it in theory. We're launching. We announced it earlier this year at the Consumer Electronics Show, but it's launching early next year. And what you'll see is firstly biological age. So the exact research in that paper that Nukhet talked about, we were able to put into an algorithm that can be run on these readings. So as soon as I get my result, I can say my biological age is 40 years old. I was born 40 years ago, but biologically the protein readings are that of someone who's 38. So that would say I'm doing something right, or my rate of aging is slower, and that's a good thing. And that's calculated by looking at all 5 of the proteins. So you see on the screen here, you would see LCN1 and ID. Those are the actual readings for this individual person. What we also do is we can go in and see the aging signs one by one. So I can say for, let's say, pores, the formation of pores or the increasing size of pores. So for pores, visually I look at you and you have no issue with pores. But when I do your protein reading that's correlated with pores, it's off. It's too high, which suggests that over time, you are more likely to develop an issue with pores. And that juxtaposition between visually, I don't see anything wrong, but biologically, there could be something worth noting. That's where we get really interesting interventions, because I can give you something now, super gentle, that's going to prevent that from ever forming, rather than waiting 5 years and you come back and you have this issue that needs a very intense treatment to correct. So that's where this becomes magic. Or the other way around, where you don't have the biological predisposition, but I do see it. So what is happening in your life, or what can I address in the moment that can help with your environment maybe? And the last area that could be interesting for us is ingredient responsiveness. So we know there are some proteins that are necessary to process certain ingredients, or related to processing certain ingredients. So we can tell you if you're a biological responder or non-responder to actives, and that would save you kind of the trial and error, the guesswork in finding the right thing for you, because I can say, no, you are going to respond really well and really quickly to this ingredient. This is the right one for you. And I can see that for sure. So that's how that works.
Awesome. A very short question. So, is there any specific age range or skin type that Cell BioPrint functions better, or does it have broader use cases?
I would say the science behind it is broad. So it's been done, it's studied on a lot of people, and it would be applicable to a lot of people. I think when it's actually used day to day, I think your medical expert, your beauty advisor, the person facilitating this would make the decision case by case whether you need this assessment, but it could work on everybody.
Mm-hmm. Oh, that's amazing. Nukhet, I mean, we have this amazing tool, Cell BioPrint. How do you think it's helping and evolving in case of skin care and aging in general? How does it pushing these boundaries and getting the science in more top position?
I would say the advantage here is to have a true personalization. It's really what it's reached. It reached the ultimate personalization. You have this kind of biological profile, and as just Fred said, what works best for you would be this kind or that kind of intervention. So really getting this very unique profile is really a help to address the right causes, first of all, and then have the right intervention for you that works for you, actually.
Amazing. Personalization is the key, I guess. Cool. So before we move on to next segment, which is future of longevity, I would really encourage everyone to send your questions to us. Would really love to hear from you, and then we can also read this question here, and then we can address them to our experts as well. So keep pouring them in. Good. So to all panelists, my next question is about the future of longevity. So let's say if we look in next 10 to 15 years, what changes do you expect to see in consumer longevity sciences? What's going to feel different in our everyday lives? Fred, do you want to answer this?
I think it's because we're starting to see it, and I think already there's cases where instead of doing a superficial solution or guesswork, you're getting a very genuine solution that's driven by longevity. So there are longevity clinics. There are more testing available, biological testing available, and people are really looking for solutions that are more intrinsic to the root cause rather than patching a solution. So in skin, can we treat, like we said, find the issue before it forms rather than do an anti-aging product at the end. So I think we're seeing it step by step.
Yeah, for sure. And maybe we can add that we can monitor also some aspects of our life and our health. And we believe it's very important to follow and to bring some solutions. Now we have some technical solutions that we do not have some years, quite years ago. So we should learn about this, and we will have more information, and we will dig into this information to find a new solution to correct the trajectory, as said by Nukhet.
Perfect. Cool. And then just again a reminder for our viewers, if you haven't yet, you can still submit your questions in our Q&A box, and then we can address them here. Maybe we could, there are already a lot of questions pouring in. That's great. Maybe we could take some of the questions from our audience. Let me get questions. Yeah. Cool. So there's one question from Rishika Agrawal, and she asked, have you mapped the different areas like protein expression? Or maybe I could do this question live so everyone can see this. So have you mapped the different areas? Does the protein expression or expression of the face differ from that of neck or back of the hand or forehead, cheeks, etc.? And does this stay consistent between the participants?
I'll give the high level answer. Maybe Nukhet, you can give the details, but in general, some proteins are more global. So it doesn't matter where you take it. It's going to be a more global expression of your biological state. And then some are a little more local. So in the process for best practice, we do left cheek, right cheek, and we keep that consistent.
Yes, exactly. I think that our more important data, and as I said, exemplified in our last clinical trial, was really done on the face. So we really like to sample where is our interest actually. So we had the clinical signs on the face, and this is why we preferred sampling the face and analyzing the biological markers on this area, on the face area. So, and yes, there is some discrepancy between legs, for instance, leg and face, mainly due to the different exposure. You have your face more exposed than your legs usually, and this is one reason why we focus on the area of interest to get the best representative samplings.
Mm-hmm. Perfect. Then we can take another question from James, and he's asking, what is the statistical power of the correlation between the markers and each specific sign of aging?
Well, I guess this is for me. So we have established a certain level of robustness on those with those data. But of course, what is interesting is to continue gathering more data to be more and more robust with time. And this is what we want to, the trajectory that we take actually to have more and more power from our studies. And maybe Fred, you want to add something on that, or no? I was thinking that you were about to say something. Sorry. So correlations, to directly answer the questions, correlations are what they are today, and they are statistically significant for sure. So we won't present and show things that are not statistically significant nor relevant, but it always can be improved and by gathering new scientific data for sure. So it's not perfect.
Okay, I think the next question is again about the Cell BioPrint, and it says from Monica, and she asked, do you use AI or ML to feed and train your Cell BioPrint tech data?
There are different types of algorithms powering it right now. So some are some more traditional, and some have done AI modeling. So yeah, it depends on the way we're doing the output and how we're looking at the data set.
Okay, perfect. Then another question is from Sierra Parker, and she's asking, the skin is very variable depending on the location of the source. It's quite dynamic organ. So how does the tool take this in account?
I think that's the question we were just answering before, right? The skin variable. Yep. So we answered that before. Yeah, we are very consistent about the sampling location being on the cheeks.
Cool. Then next question we can take from Josh, and he's asking about skin aging. Have you seen the impressive result by the new One Skin product like OS1? Does L'Oreal have similar products? What are the comparable creams, etc.?
I'm not sure about that product. Do you guys know?
No.
No, unfortunately.
Oh, I'm sorry.
Okay, sorry, Joes. Maybe you can give us a more detail about OS1 and then could help our experts to make a comparison. Then next question we can take from Ashish Rajput, and he's asking, can you please elaborate on research activities happening at L'Oreal? And if you could give us a bit of an idea around it.
The research activities, you said?
Yes.
Well, we are working on many actives and many well-known actives also, but we are still searching and continuously searching the perfect active that can fulfill all the performance we want to tackle for longevity. That is for the technologies. And for the research activities, we are working, as said, on the hallmarks you have seen, on the different mechanism of action, and trying to understand the interconnection between hallmarks, because it's so complicated that I think we will need many years to tackle this.
Definitely. I think the next question is also quite interesting, and Monica is asking, there's also a rising awareness about implementing a skin routine for men. Do you have any similar data in a men population?
Yeah, this is a very good question, actually, and it is the part of the data we lack, actually, because mainly our clinical trials are done on the women, and for sure, because of this trend, men get more and more interested in their skin, and we need to gather more data now from men to be at the same level, I would say, of precision than we are today on women.
Mm-hmm. Perfect. Then maybe we can have some additional questions on Cell BioPrint, like how can we get more information about the Cell BioPrint? How can we learn more about it? Is there any supplement information or where we can look for it?
I think for this, we did a big announcement at the Consumer Electronics Show in January of this year. So actually, you can Google this one. And there's a ton of walkthroughs of how the experience works, some kind of explanations about the science behind it. It was written about in a lot of publications, and there's a L'Oreal press release. So you should be able to find a lot. And if that's an internal question, we can also provide that internally as well.
Perfect. We have one more question, and this is about more, I think, in general, the skin and different metabolic conditions and gut health. And it says, Andrew asked, have you looked at the gut health and its impact on skin conditions and ultimately longevity?
Not so far for my understanding of the question, but for sure we know that we have a very strong connection and very strong insight from the gut microbiome. In fact, all the microbiome we developed internally here for the cutaneous microbiome is well inspired by the gut, for sure. But I really think that the gut health and the microbiome could have an real impact on the longevity field. That's why in a longevity, globally speaking, broad approach, in fact, supplements could be very helpful to that. And we believe in that because, as you know, the gut microflora could bring something directly, finally, into the blood, while it's so complicated for cosmetic, for topical application. So you can have a global impact on all the organism and not only of the skin.
Mm-hmm.
That's why it's so interesting for longevity by itself in general, speaking human health.
Perfect. I think the next question from Leo is about more supplements, and he's asking, novel supplements offer human identical compounds like creatine, collagen produced in plant, show superior biocompatibility, transdermal absorption. What does the panel think the commercial potential of such product? Is any of the product that panel would like to see?
So maybe I can start. As I said, there are many clinical trials that are ongoing on the effect of those supplements and on the objectivation of the effect of those supplements in health. But data is lacking on skin, actually. So there is a real need of evidence, again, to make the demonstration of their effectiveness. So this is what we need to tackle and bring objectivation on that.
Yeah, for sure. And I think it's, as a consumer, I think it's a super interesting area. And I think in one spot, I think for like advanced glycation and products and their role in longevity, I think we see some interesting work with supplements there. One of our brands, Skin Better Science, just launched a supplement in this space actually, and it has great results. So maybe look at the Skin Better Science supplement.
Mm-hmm. Perfect. Then Natalie is asking the more applicability for Cell BioPrint. She's asking, can the Cell BioPrint, in addition to detect responsiveness, can detect ingredient that user may be allergic or not tolerate to?
I think I would say it's focused on proteomics, so measuring protein levels. I don't know that, is there a direct correlation between that and allergies? I don't think so. I don't think we'd use this particular technology for that.
No.
No, we're talking more about the direct responsiveness of like if your body will be able to use that ingredient.
Perfect. Then we can take a few more questions. And Eva Dodson is asking, what are the opportunities for working with biotech startups using your technology at a startup level? So I think more collaboration kind of approaches that what the Cell BioPrint can offer.
Yeah, I think we love collaborations. I mean, we have a few thousand internal researchers creating a ton of new ingredients and intellectual property and research, but we also love to work with the outside world. So we have an entire team called Open Innovation that finds startups and does these partnerships. I would say even for this project, Cell BioPrint has a strong partner called Nukhet, which is a company in Korea that is a leader in diagnostic tech that we work with. So yeah, we're always open to this.
Cool. Then maybe we can take a few more last questions. I think this question is more about having more biomarkers. And Megha Shah is asking, when you speak of integrative approaches, have you seen non-skin related biomarkers to predict skin aging, like metabolic marker? Something I think Nukhet was already talking about.
Yeah. So we looked at our proteins, our skin proteins, and when you work with all those proteins, because from one sample you have thousands of proteins actually from one sample of skin, and they don't have all a specific function known in skin actually, but can have a different functions that are not specific to skin. So yes, we can either get inspired from their other function in other organ, or make the demonstration what is the function of this specific protein within the skin context. So yeah, it happens.
Okay. Maybe one last question I can read is, dear L'Oreal experts, it's from Hannes, and she's asking, what can we as a general longevity community do to support work in the field, beside direct featuring your product at event, etc., what you're looking for?
In my experience, I know we are very plugged into the community. We have key dermatology, doctor, researcher, and clinician partners in the longevity space that we work with day to day. So I think those are kind of very specific relationships, one-off, and we trust these experts with helping guide some of the work that we do. I think we're also very present at panels. We're very present at conferences, which is like the American Academy of Dermatology. So we'd love to engage more and kind of explain what we're doing and get feedback all the time. So I'd say that's from my perspective and a broad approach.
Yeah, you're right. And what we want to bring also is to contribute to the search, globally speaking, to the scientific research, to bring some clues, some evidence that we have some big rationale behind, in fact, the mechanism of action we use in our products. The product, for sure, we want to sell some products, but it's always kept backed up by some scientific evidence, proof of evidence, and Cell BioPrint is for us a way to show this evidence. But behind, we have a lot of science and a lot of years of science, and maybe Nukhet can talk about that, but it's very hard to find the right biomarkers, how we can detect them, did they change or not during aging or in some pathological conditions. So we have plenty of subjects of concerns.
Yes, and we like publishing this science actually. So publishing is really to to be confronted to our other scientists, our peers, and because we are reviewed by them, first of all. And so it's a way for us to participate at our level to the global scientific community, depending on the topic. But here, the same for longevity, I hope, and more and more.
Great. Perfect. I think now we have a few more questions, but maybe we can answer them later because we're also approaching the time. And with this, I really want to thank to our panel and also to all of us for tuning in and asking these great questions. So today, with our panel, we talked how science is helping us not just to live longer, but live better with healthier skin, deeper biological understanding, and tools like Cell BioPrint that could change the game in how we care for ourselves. So these are some of the summary points that we hear is what longevity really means, the technologies and biomarkers shaping the space, and where the future might be headed. I think more personal, more preventive, and more science-based. And with that, thank you so much again, Carine, Nukhet, and Fred. Thank you so much, all of you. It's been a great discussion. And then take care, and then see you all next time. Thank you so much again for joining in.
Thank you so much, Amrita.
Thank you. It was a pleasure.
Yeah.
Bye-bye.
See you. Bye-bye.